titled Illuminating Functions of the Understudied Druggable Genome, or the Druggable Kinome.
All right, thank you very much for the opportunity to talk to you today. As Stefan said, I'm in biomedical engineering and pharmacology at UNC Chapel Hill. And my group's one of 6 that are part of this IDG project that Mike McManus just described earlier. And again, we are what are called as a data and resource generating center. So the groups involved include Gary Johnson, who's in Department of Pharmacology at UNC and has worked on kinases for decades. So he's really a core person for this and is the lead PI for the whole group. Tim Willson, who's in School of Pharmacy at Chapel Hill as well, a chemist, and is also a big part of the SGC. Peter Sorger, who's in the Department of Systems Biology at Harvard, is involved as well, along with Ben Major, who a few months ago was at UNC Chapel Hill and has just recently moved to WashU, along with Reed Townsend, who's both are very experts or in-depth people at proteomics. Reed Townsend in particular is very much focused on the technologies around quantitative proteomics. And so together, our focus is really around a very protein-centric view of the kinome. So while we certainly do lots of sequencing and CRISPR and those sorts of things, not at the scale as just talked about by Fiona, that we have a pretty heavy proteomics view of the kinome. We're very interested in drugs and basically how they work on proteins. So just a quick intro to me and my group. We're a systems biology group, primarily computational. We have some experimental efforts that are targeted at very specific lines around validation or specific areas of interest. We are broadly interested in developing tools and technologies to better understand the behavior of biological systems and hopefully be able to manipulate them. We do a lot in cancer and infectious disease, and our computational work spans everything from sort of detailed mechanistic models, you can imagine ordinary differential equations, and Michaelis-Menten sort of types of large-scale modeling and simulation, up to more machine learning, probabilistic, deep learning types of approaches, depending on the type of data we work with. And just big picture, again, with the IDG and these data and resource generating centers is really it's to try and improve our understanding of the properties and functions of these largely unannotated but highly druggable protein families. And as Mike had mentioned, it's focused on GPCR as ion channels, and our group is the protein kinase setup. While a lot of the work that we have done and is being done in cancer and cancer models, it is supposed to be disease agnostic, so we don't care. We actually want it to be as broad as possible as far as understanding where these proteins may play a significant role. And a major component or driver of this is to generate and distribute data, resources, tools, annotation to the broader research community. So that is a central goal, and in fact, probably from NIH's standpoint, a major driver is to enable others, in particular thinking of other investigators, to write R01 or equivalent scale grants on these understudied proteins by building on the resources that we're helping and others are trying to develop so that they could pursue these targets where maybe there wasn't enough previous data to really investigate them further. So just a little background on the kinases that we're particularly focused on. There's 162 kinases that are on this list coming from NIH out of about 520. And just a little background and context on average, a cell will express about 350 of these kinases in general at one time, with roughly a third or so of these being this understudied category. So they're by no means rare by any stretch of the imagination. They're there. They just haven't really come up at the top of list in a lot of these earlier studies. And they are, with the exception of the TK family, which has a single understudied LTK member in it, The other understudied kinases are fully, fairly uniformly distributed across other members of the kinome tree. So one of our fundamental principles or approaches, I guess, is that we look at kinases in context, and this will hopefully make a little sense in a second. We look at kinases, understudied as well as well-studied kinases are working as a system is how we approach it, right? And they are integrated into sort of these functional subnetworks. And there's significant crosstalk and communication between these kinases and the signaling pathways which they form, which leads to significant amounts of resilience and adaptability, and where we see it a lot in cancer is obviously in terms of reprogramming and adaptation to response. And so this is kind of our little toy model for a lot of the therapy approaches that are used, right, where you use a drug, you hit a kinase or your targets, and it works, or some population maybe works, but other members of this pathway or kinases or even other members of the non-kinases will help adapt the cell to it, and you get drug resistance. And obviously this is where some opportunities for combination therapy and so on come about. But we are very interested in looking at sort of how these things work together. So while we do have a focus on understudied kinases, we look at all kinases basically and how they work together. So I'm going to give a sort of a whirlwind tour of some of the work that we're doing, a brief overview of some of the work being done by the different groups. I'll highlight a little bit of some of the systems aspects, a little bit more detail on some particular project that we have going on, and a tie-in to this knowledge base where we're a working prototype in a sense, where we're providing all the data and trying to provide different views of how to interact with it and predictions and so on. So just well, briefly, Ben Major's group is really focused around the establishment or identification of protein networks around each of these understudied kinases. So a lot of flag and mini turbo sort of based approaches for tagging basically potential ligands to or interactors with a given target. So the idea, this is also tied with, say, more functional screens, say with CRISPR-I or CRISPR-A. And the end result is really supposed to be these essentially maps of, for each understudied kinase, what are the sort of surrounding sphere of interactors that it works with, right? So we are doing this for all of the 162 that we have on our list. Tim Willson's group is really focused on sort of exploring some existing or trying to identify chemical tools that could potentially be of value. So this is following the SGC sort of guidelines for what a tool compound essentially should look like. And so a lot of this is the establishment of nanobret assays where we are basically, and some of this does go into, say, KinomeScan or DiscoverX, where we're trying to look at, identify a subset, some compounds that do have some degree of potency, some degree of selectivity that could have the potential to maybe be used as a starting point for exploring some of these understudied kinases. To date, I think there's 39, it may be closer to in the 40s now, that of the kinases that have at least some starter seed set for this, and the associated information we're also generating and providing for anyone. Peter Sorger's group is involved in a lot of things, but this is just one of them. There's technologies such as what's called SciSif, this dye drop one, where basically he's doing a lot on the phenotypic screens, basically. So we're doing perturbations of various sorts, whether it's CRISPR or drugs, and measuring phenotypes. In this case, it's through a dye drop assay where cells are actually put into wells, the 384 well plate, and we can stain for different markers and make different sort of assays, and this is done in a sequential iterative process, so where you can basically multiplexed readouts of a sample in response to a certain perturbation at given points in time. And then, again, this is tied with another type of sequencing, a little bit lower cost, but allows us to screen lots of samples with a lower degree of coverage, but it gives us a better picture of what's going on in these cells. So basically a lot of phenotypic screens of different sorts are also in this effort. And then I've kind of lumped Gary Johnson and Reed Townsend and myself into this quantitative Kinome Dynamics, and I'll give a little more detail, sort of a vignette on some of the work that we're doing with this. But the fundamental idea, again, it's a very protein-centric version, but we'll provide cellular perturbations. And this basically goes to this centerpiece there, is this column. And we're using a technology, which I'll explain a little bit more in a second, called MIB-MS. So it's multiplex inhibitor beads with mass spectroscopy. It's very similar to Kinobead setup, where essentially we have specific compounds covalently linked to cephalose beads layered on a column. We can take a sample before or after a perturbation. It works with biopsies as well, so we've done this with needle biopsies in breast cancer patients and other biopsy situations. And we can lyse the cells, run it across the column. We're basically selecting kinases preferentially in their active form, so we're looking sort of at an activity states of the kinome. And then those run through mass spec, and we can look at, essentially get a profile, a signature of what the kinome looks at that point in time. With Reed Townsend's group, so we're doing a lot of parallel reaction monitoring. So what we have actually, I think, in hand now is unique peptides for all 162 kinases. There may be one kinase that isn't quite finished yet, where we have a unique peptide that flies well on the mass spec that essentially you can do the equivalent of a Western, right? A Western blot with mass spec. So we can do very atomol level, but I know I've seen femtomol quantification of kinases in the samples. So we can, and these are for, kinases that don't have antibodies, for instance. So we can do quantification of how much protein is in the sample through the mass spec route. And so this, again, looking at different perturbations and how this works through this dynamics, then we go more towards sort of the modeling and analysis and interpretation phase, which my group and Peter's group as well are pretty involved in, and as well as sort of the data integration and providing that through out to the broader public. And so I'll touch on it at the end. We have what I call a working prototype website. So it won't really work on your phone, so it's not mobile friendly yet. But essentially it's a resource for us to, one, make data and available, as well as some tools. And we do a lot of working with people who actually are interested in this area and then trying to develop specific views or things that would actually be useful to them. So I'll touch on that at the end here. But so just I wanted to do a little bit more of a more detailed example of what we're doing with the MIB-MS work. So this is an example where, again, we have these beads, covalently linked compounds, kinase inhibitors that are in a certain combination that allows us to basically capture a very broad range of the kinome. And so in this work, we looked at different, in this case, it was cell lines, clonal and basal-like, right, which together are sort of fall into the triple negative breast cancer batch, HER2 and luminal. And together we can capture nearly 90% of the expressed kinome with this bead drug combination. So over 400 of the kinases are observable through this approach. And in this particular work, we looked at just 15 breast cancer cell lines, replicates, and again, representing all these major subtypes. And we did a MIB binding profile for each sample, basically. And on the right, you can see sort of, again, that each row represents a kinase, and the columns represent the different subtypes. And you can see the red and blue block is sort of together, and that does represent the triple negative group of cell lines. The pink is this HER2, and the luminal is the black. And those all partition very nicely based on the signature of this activity profile, essentially, of each of these kinases. And these readily partition, separate into the different groups with again, the basal, claudin, HER2, and luminal. And so this was a work that we had done to really just get a feel for how much information was in these profiles, how distinct they were, how predictive they were, in particular in first pass of just discerning subtype. And so one of the things we've looked at is we looked at different features, different kinases that were highly predictive of subtypes. So we identified a set of 50 of what we call pan and subtype specific kinases. So there are certain kinases that you could imagine are predictive across all the kinases, right? So for instance, if it's very highly present and active in one cell type and less in another and less in another subtype, right? That would give you, if you measured that, it gives you some information maybe across some subtypes. While another one, say for instance HER2, right, is clearly a marker for HER2 positive, HER2 enriched breast cancer, but just it doesn't tell you much about any of the other subtypes, right? So if you put together these sort of markers, we were able to make a very predictive, essentially signature of a given subtype of cancer. And what was interesting in this is about nearly half of the kinases that are part of the signature are considered understudied kinases. So what this told us a lot, for one, is that even though many of these we have some understudied kinases, but by no means are they not playing a significant role. In fact, their status, their states, is highly predictive of, at least in this case, a subtype, but we've also seen, I'll touch on in a second, but how it also responds to drugs. So, we looked also at response to different drugs. So for instance, the SUM159, that's a triple negative breast cancer subtype, and the HCC 1806, that's a basal. So claudin low is a SUM159, the HCC is a basal, so together sort of this, what's normally considered triple negative. And they were both given the same amount of trametinib, which is a MEK1/2 inhibitor, and what you could see is that there was a very different response, dynamic response in the kinome sort of profiles, how strongly they responded to the same dose of drug. So this is an example, right, where the kinome responded very dynamically in response to these targeted kinase inhibitors, but this triple negative subtype, and you can look at it, two components of that, right, two versions, actually have very different responses. And if we look at subnetworks, which I'll talk on in just a minute here, those individual subnetworks also responded very differently and dynamically to the same sort of input perturbation. And again, with the green dots here on the left just show that it's sort of the magnitude of change of these kinases. All the green ones are these understudied kinases. So the understudied kinome is very dynamic, very responsive to drugs, and clearly has reason to be studied, partly because it really does look like there's opportunities for both identifying targets, and as they are kinases, we do have the potential to actually develop tools and drugs that could be involved in manipulating their behavior. So one of our central issues, right, questions is how do they work together? Where do understudied kinases as a whole fit into this? And so what I want to touch on briefly here is what is the architecture of the kinome? So for us, again, we're looking at this very much from a system standpoint and not as a single target, but how does this target fit into the broader context of who it talks to? And so one of the things that we've been looking at is basically big networks of kinases. So we've looked here, I'll just talk a little bit about this work where we took different databases, basically, protein interaction, phosphorylation, other sorts of data to just assemble a protein interaction network, which, when it was all sort of massaged together, was about 5,000 edges among these about 500 kinases, right? So this is just the kinome, just kinases. We aren't looking at other non-kinases. And What is also relevant for these downstream studies and is required actually is that we look at a weighted graph. So it's not just that the interaction's there, but there is a value, there's weights just sort of describing the strength of interaction between two kinases. And this was built up by looking at co-occurrence in KEGG pathways, and I'm happy to talk about that later. But basically it provides a functional weighted graph that then feeds into downstream algorithms. And so this is, you know, the messy ball, hairball thing that comes out of that. So it's a very, there's a lot of things going on here, right? And so what we're very interested in is, more broadly speaking, you know, what is the organization? Is there a way we can look at this that provides a little bit more of an organizing concept behind it? And so what we have done is a lot of what's called community detection in certain fields, identification of subnetworks, clustering, graph clustering, with the goal here of really trying to identify communities of kinases that are working together, at least in this context. And so we've looked at a number of different algorithms, methods for this. We've developed some of our own on the side. But just for now, we're talking about this group here. And one of the things that's commonly done is to, when you do this clustering, this graph thing, is a common metric is what's called a modularity score. And this is fine. This is used all the time to identify sort of when you sort of have mathematically a good set of definition of what independent sort of clusters look like, right? Subnetworks. The nice thing, the key about that is it's a mathematical definition, and it doesn't necessarily have anything to do with the biology. There is, surprisingly, actually a fair amount of agreement between that. When you see things that have good modularity, that often looks like that's not too far off biologically, but we are very interested in having independent ways of sort of investigating that. And that's where we have what's called, and I don't have time really to talk about it today, but we're a method called sort of learnability, right? Which is both of these blue things. One's a multi-class score, which is just like it's essentially a correlation coefficient, but allows you to deal with highly unbalanced data, but it's using our learnability matrix. And essentially what this is we are using I think I have it here, is an independent set of data. So you could imagine basically given this network architecture, right? So each of these methods provides a different sort of picture of how things are structured and organized, right? Different subnetworks. And you say, well, given based on this protein interaction and phosphorylation and other data sets. So based on that, if I have, for instance, gene ontology or some other pseudo-independent data, right? How well does this data, if I were predicting a set of clusterings from that, how well does this set of data actually predict that clustering, right? So it's not gene enrichment or gene set enrichment analysis or any of these other sort of methods. It's which doesn't really apply well here. It's a different one where we're essentially using another learning algorithm to try and say, if you gave me this, how well would this data actually predict that? How well do those things agree with each other? And what we found actually is that this Louvain method, which I guess it's not showing up here, we'll go to it, is the one that seems to really provide the best sort of, it has very good sort of mathematical modularity, but it also has the best sort of learnability from other sorts of data methods. So we can use other machine learning algorithms to try and predict these network structures. And what it looks like is this here. So this is the kinome as a whole, and you have these big circles, which are superclusters. And within those superclusters, you have subclusters. And again, the green represents the understudied kinases. And so we see, I believe there's 9 of these superclusters and 26 of the subclusters. And one thing to note is that these understudied kinases are distributed throughout these subnetworks, right? And one of the reasons we think the MIB-MS is working quite well with a small set of measuring, if you measure a subset of kinases, right? If you look at where those kinases are distributed in these subnetworks, they're essentially almost all subnetworks have a representative in that. So you could kind of imagine, like, as an engineer, say an oscilloscope or something like that, but if you have a probe at each of these spots, right? You can't measure everything, but if you measure a few members out of each one, you get a rough approximation for what's going on, which is fairly predictive, and that's how we're thinking, or why we think some of these signatures that deal with a subset of things, those features we're measuring sort of at certain places in these subnetworks that have fairly predictive value, and so we don't have to measure every single thing to be able to make some of these predictions. And just to sort of blow up right is that you can see we just have these subclusters, and those, again, the learnability part is using other machine learning algorithms to basically say how cohesive, how well would we have predict this architecture, right, given gene ontology data or some other data set, right? How well does that agree? So we've found that that's actually been a really useful tool that we want to use in several ways. And one thing I just want to bring up as well as we talk about dark kinases in terms of sort of focus very much on the node itself, on the kinase, right? So how, if you know, this is understudied, that could be because this doesn't have, we don't have any antibodies for it. If you knock it out, it has no obvious functional phenotype, right? Or phenotype, maybe because other kinases sort of fill in the gap when you knock out this one. There's all sorts of reasons why this could be understudied and dark. Another component, though, is even though we know it's there, and even for some of these, quote, well-studied ones, what happens if we gain new information about a relationship between two things here? So, you know, I think of it a little bit, at least from the US, right? What if there was some unknown relationship between Obama and Trump, right? That would be kind of shocking. And the same sort of thing here. What if we find new information? How much of an impact does that change or bring to how we view the organization of the kinome? So this is just a different way to look at how the kinome's organized. In some ways, it's a sensitivity to noise tests. So for instance, how robust is the subclustering, the network that we've identified. But it's also another view of what's the impact of if we find new information that could potentially, how strongly could that alter our view of how the kinome's organized. And so a simple way to measure this is through what's called the adjusted RAND index. So if it's 1, it's then you did a change, you put an edge in, and it did nothing. It changed the subnetwork structure, didn't change at all. And 0, it could be just this, it's completely different in every possible way. And so when we have looking at that is a plot basically where we have the change likelihood. How likely is an edge to introduce a change? And if it does introduce a change, what is the magnitude of that? And so you could imagine that if you're up in the upper left, it's unlikely to change. And even if it did change when I introduced this edge, the impact, how strongly it changed the subnetwork structure would be very little. Versus the right-hand side, if most changes I put between this node and something else are very likely to cause a change, and when that change happens, it really changes the structure of the organization, the kinome. And what you see is, for the most part, right, to just, most things are along this unlikely to change, little impact, or likely to change, little impact. So the upper right, this basically is sets of things that it is likely that if I introduce new information about this kinase and maybe what other kinase it talks to, it does change things, but not by a lot. But then there are some other things that actually, if you introduce edges between them, are very significant changes. And things like node degree, like how tightly connected and other things a kinase is to another one, actually is not predictive really at all for how this works. And so what we see, largely speaking, is that, again, understudied kinases are distributed throughout this. One of the biggest ones is actually ReOK3, an understudied one that if you add new information about what that talks to, it actually can cause significant changes in the way things are organized. And so it's just we're looking at this as another way to understand sort of unknown relationships, and interpretation of other sorts of data, interaction data, and other things that we are pulling together. So This is an example of, on the left is the sort of what we consider our baseline architecture, and on the right is one where one of the biggest changes, actually, if you introduce a single edge, I believe it's between MapK11 and FGR, it significantly changes the way the structure of the kinome is structured. Again, it doesn't mean that actually these things are true, right? Just because you put an interaction doesn't mean that it actually exists or happens in the real world. It's just, it gives us a measure of how sensitive certain places are to new information and how you can interpret that. So this is kind of a teaser because I don't really have time to go into that, but one of the things I think I touched on before a little bit was this idea of this combination therapy thing. And so what we're looking a lot at is, clinically, many of the more sort of dirty drugs are actually more effective. And a lot of that, at least clinically thinking, is that, yes, I'm hitting my given target, but I'm also hitting other things that help stop reprogramming and resistance from happening. And so what we're doing a lot, you could imagine that as a first pass, right, each node in this is a variable in a nonlinear regression model, essentially, right? As well as the subnetworks, right? So groups of kinases that sort of work together are another variable of their own little coefficient, right? And if I link this to some phenotype like growth or some other behavior, right, I can use the states that I can measure each of these nodes and make predictions about what the output behavior of the kinome is. And if that state of the kinome is linked to something like growth, I can start to make actually predictions based on the state of the kinome. And so we have other work where it's more sort of on the machine learning side. I can't say it's really deep learning because there's not enough data for that, but where you're looking at embeddings, you're looking at vectors of kinase states that represent some high dimensional space, and you can then start to map out what that space looks like and link it with phenotypes. And then if you can move your system by using drugs to manipulate the kinome to get to a certain place in that, you would predict that you would get the outcome of what that phenotype is associated with in that space. So that's fairly abstract, but the idea is to actually start to bring some of this to a more practical, a testable place where we can actually start to use drugs to manipulate the system. So dark kinases are a huge part of this. They're really no different than the other kinases in terms of the degree of magnitude that they play in the behavior of these systems. So we think this is a really nice opportunity to learn more about where understudied kinases really can have a role and have an impact as a target for therapy. So all of this is going into what we call essentially is a working prototype, this darkkinome.org. We are in the process of providing all the data, for instance, all the PRM peptides, all those, as well as the forward and reverse curves. So if you wanted to do your own PRM experiments, here's the peptide that you need, here's the concentration, and you can do quantitative proteomics with this. We have a host of tools that we're starting to put in. That's kind of more our orientation right now, is to develop tools that provide views to the data. So we have things like Indra, which is actually mines text and provides relationships between whatever you search, but in this case we're interested in kinases. There's a small molecule suite, which is a set of tools to sort of explore small molecules and what potential good targets they have, especially in the dark kinome space. We are working on sort of a browser. We've had lots of people who have asked us, well, I just want to see in the context of kinases only how this kinase rates to other kinases in terms of expression or protein level within a given tissue. There's a lot of interest that with people we've worked with, that I want to know about this particular organ, what's there, what's highly expressed, how's it relative to this one, disease systems, and so on. So we're trying to develop tools in this way. And so one, we're very interested in use cases. So if you would be interested in getting having us show you something or provide some view of what this or other data looks like, please let us know, because we will try and do something. Right now, compound data appear on peptides. We have CRISPR guide sequences because we've done our own CRISPR-I screens, so dropout screens to look at important kinases, nanobret data, interaction data. All of that is being funneled onto this site. So with that, I'd like to thank you very much for your time and for IDG for funding this. And thank you.
Thank you, Shawn.